Hello friends, this is Miss D.A. Marvekar, working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. And in this very small and interesting video, we are going to study very interesting topic, that is DNA fingerprinting. This technique is used in the forensic lab to resolve the parental dispute, to resolve the murder cases. Now, like Aadhaar card, during Aadhaar card formation, we have given our fingerprints. Why? Because that are very unique to ourselves only. So similarly, certain DNA sequences which are present in on our DNA that are very unique to us. Even that will differ in the twins also by the single nucleotide. So definitely we can differentiate between the twins. We can also identify the criminals if we get that particular sample from the criminal side. This technique, the credit to this technique goes to Sir Alex Jeffrey, who invented this technique, and he realized that we can use this technique to detect the variations in the human DNA which are present in the form of a mini satellite. There are different cases which are present in the history of a mankind which has resolved by this great technique. This technique has resolved the parental dispute, murder cases and the rape cases as well. I will tell you one unique case which has resolved by this DNA fingerprinting technique. And this case was of Naina Sani and her husband Sushil Sharma. Both were active in the politics, and after marriage, he killed her wife and he shot her wife and he took her body so that he can dispose her body. So he took her body and tried to dispose at the restaurant called Bagia, and after with the help of a restaurant manager, that is a Keshav Kumar. The body was put in the clay or the tandoor as we use for the tandoor chicken, and he burned her body, and they both managed to flee from the police. So after certain duration, he surrendered to the police, but police were investigating what was that, or that body was of whom. So definitely, police took the help of this technique. They took the help of a forensic expert, and when that burn sample of the body was taken into the forensic lab, that was detected that body was nothing but of the Naina. This is a very famous case as a present in the history as a tandoori case. For now we will study the different steps required for the DNA fingerprinting. The first step is the collection of a sample. Whatever criminal has happened, we will obtain the DNA sample from any of the body tissue or the any of the body fluid. I can use the buccal smear, I can use the saliva, I can use the tissue, I can use the amniotic fluid, etc. Once I will isolate the DNA, when I will collect the sample, I will isolate the DNA, I will extract the DNA from the collected sample. So obviously if the sample is very small, the DNA which we will isolate or extract will get a very small in the amount. The technique of isolation or the extraction of a DNA will differ from the tissue to tissue. So there are different types of methods are there which can be used suitable to the suitable tissue. There is a phenol chloroform method is there. You can use the CTAB DNA extraction method. We can use the proteinase K DNA extraction method. Once I will extract the DNA, so definitely in order to multiply the DNA copies, I will prefer first the restriction digestion, and then I will amplify the DNA fragments with the help of a polymerase chain reaction. Once I will get the multiple copies of a DNA, I will proceed for the DNA isolation, DNA separation with the help of a agarose gel electrophoresis. Here, in this agarose gel electrophoresis, the DNA fragments present in the respected sample will get separated according to their size. Here, during investigation purpose, whatever the sample we have collected from the evidence side and whatever the suspicious individuals are present, their sample I will isolate separately, and that much amount of a sample I will load into the different wells on the gel, and then I will perform the agar gel electrophoresis. Now each DNA fragments will run according to its molecular size, so definitely this pattern I will get. Whatever samples I will be, there will be there. I will perform the same technique for each and every sample. After performing electrophoresis, I will next go to the Southern blotting technique, in which I will transfer the DNA separated on the gel onto the nitrocellulose membrane. Once I will blot that DNA fragments on the nitrocellulose membrane, then I will hybridize the DNA which are present on that membrane with the help of DNA probes. Now, these probes are artificially synthesized oligonucleotide chain which can be radiolabeled and which are very unique or very specific to the specific DNA fragments. So, when I will add the radioactive probe on that membrane, the probe will bind to the specific site. After hybridization, I will just take the X-ray film. I will pass the X-ray from the DNA fragments. So this I will get this pattern. Definitely I will visualize this DNA bands and I will interpret the result. During interpreting the result, we can use different types of techniques. The DNA fragments which are transferred on the nitrocellulose membrane and southern blotting, we can during analysis we can use two types of analysis. One is a PCR based analysis and the second is a RFLP based STR analysis. Now the restriction fragment length polymorphism, it was the first method which was used sorry, for DNA fingerprinting, where the restriction nuclease enzyme sorry, which will cleave the DNA in various sites and separated on the gel. In the PCR based analysis, which is very popular one and which is a very simple than the RFLP based method, here we will get the reliable result during interpretation. Now suppose this is the sample which we have isolated from the crime scene, and these are the three individuals on which we are having a doubt that they were present at the site of a crime. So I will take DNA sample from each one of them and then I will perform this technique. I will extract the DNA sample, I will strictly digest that sample, I will perform the PCR and then I will go to the southern plotting technique and then after hybridization, I will interpret the result. If the DNA bands present in this sample will get matched with this individual, so definitely it will indicate that this is the suspect which was present at the scene of a crime. So there are different applications of this technique, that is to solve the parental dispute. We can identify or test the paternity and the maternity. We can identify the criminals. We can also analyze the phylogenetic analysis of any plant samples or the animal samples. We can also test the food if which is contaminated with the different types of meat, and we can also identify the crops. Now we can also solve the parental dispute. Here suppose these are the three dads which are fighting with each other and they are telling that this is their child. So definitely if this case will go to the forensic labs experts, so definitely they will take the DNA sample from each one of them along with this small child and their and his mother. So after performing the DNA fingerprinting technique, and when they will interpret the result, they will check that whatever DNA fragments are present on the child, some of the DNA fragments will match with its original dad and some of the fragments will match with his mother. So definitely the fragments which are matching, some of the fragments which are matching with any of the dad, so definitely this dad 3 is his original parent, and there is no any challenge for this result. This will prove that the parental dispute will get solved easily. So I hope that you have understand this technique very well. Thank you so much.